All righty. Good morning, everybody. This is the Lab Analyst grades 1 through 4 TCP review. I wanted to just start by giving out some big thank yous. Our first thank you is to Shannon Simmers because she was the one to reach out and get this whole event organized. So thank you to Shannon. And then I also wanted to give a huge thank you to our trainer, Katie King. She's volunteered her time and energy and talent to host this training for all of you who are here in person and all of you who might be listening to this as a recording. So I wanted to also say a very big thank you to Katie. We appreciate you very much.
Thank you.
With that, I'll just pass it on over to Katie. If at any point you need me, I'm going to be sitting here monitoring all the webinar stuff. So just shout out my name if you need me. But otherwise, Katie, take it away. Thank you.
Okay, thank you, Amanda. All right. So I'll just give you a little bit of background about myself. Like I was telling Rich, I graduated from Cal Poly San Luis Obispo with a BS in biochemistry. And at the same time, I was in the Army to get through college. So that helped me pay through college. So I did some time there. And then when I got out of college, I went into cancer research, and that was fun in New Mexico. But I wanted to come back to California because I love to surf. So I was lucky enough to be able to come back to California, get a job, and basically I'm working where I pretty much grew up. So I feel real fortunate. I work for the city of Scotts Valley wastewater, and we have a reclamation filter plant. So that's kind of where I'm at, and pretty happy. But with my experience with the study sessions is I went to the study session 2 when I was starting out because I wanted to just skip the 1 and try to go for the 2. But I found that it was too much like the study guide. And I was kind of looking for maybe a little bit more of like, okay, what's really going to be on the test, or, something other than the study guide, which I could kind of get on my own. So I spent years talking to people, and after I completed my last certificate, the Lab Analyst 4, I thought, I think I got some pretty good ideas of how to help people out who are, getting ready to test for their certifications. So I put this slideshow together. Actually, I really tried to condense it and cater to everybody. Do we have any like lab analyst ones that are starting out, or anybody studying for the lab analyst 1, or is it mainly?
Yeah.
Okay, well, good. So I tried to cover everything for 1s, 2s, 3s, and 4s. So there's, and the 3 and the 4 it is a combination of the 1 and the 2. So you never get away from it. And so I think I did a good job covering everything. I hope. You guys will have to let me know. And but it is condensed, so I'll see how that goes. We'll see how it goes. But so for that's pretty much my background, and I'm hoping I can, help you guys out today. And then I don't know if you guys want to kind of introduce yourselves and let me know what you're studying, what you're going to try and exam you're going to study for, or if you just want to start.
Okay, Jen, do you want that information? We're open to either one.
Shannon, do you want to try to introduce everybody at your place, and then?
Sure. Yeah, okay, so I'm Shannon Simmers with the City of Redlands Regulatory Compliance Officer, and I'm looking to take my grade 1 laboratory analysis exam so that I can be more helpful to my laboratory people here to grow professionally. Audrey?
My name is Audrey. I'm a lab tech here at City of Redlands, and I currently have my grade 1, and I'm looking to get my grade 2 in September.
All right.
Hi, I'm Cwea, grade 2 analyst for City of Redlands, and I'm planning to take my grade 3 in October.
Okay. Good.
Hi, I'm a grade 3 analyst for City of Redlands, and I'm planning to take my grade 4 in the summer session.
All right. Last one. Good. Get that 4.
One.
I'm Fishling, what manager here at the Redlands Wastewater Treatment Plant. I want to take my grade 4, which is a job requirement. And when I took the grade 4, I think by 4 points.
Okay. So you have kind of an idea of what to expect. That's good.
I have an idea of what to expect. If they haven't, now when I took it, you had to drive somewhere and break out a pencil and a calculator. So here I hear it's at a Sylvan Learning Center. It's all by computer. And so it's going to be a big adjustment, yeah.
Okay. All right. And then
JT's not here yet. So,
John thinks he's going to plan to take the grade 4.
okay.
He's
So we have,
he's currently a grade 1, but he has 5 years experience, and he's going to skip the 2 and 3 and go to the 4.
all right. I admire that. That's good. That's good. Okay. And then Rich, did we hear from you?
Yeah, I kind of introduced myself right at the beginning, but Rich Novick, I'm a lab analyst at the City of San Luis Obispo and have my grade 2, been there for 4 years now, had trouble with the grade 3. And like you said, the study guide basically guides you to memorize the whole standard methods book. And for me, that's kind of difficult. And I like your idea of maybe a narrowed focus on the test.
Yeah, I'm the same way. Standard method, it's like overbearing. It's too much legal jumbo. I almost go to sleep when I have to crack out the standard methods book. So, yeah, and in fact, a lot of times I would just go to the internet and get like a condensed, if I had questions, trying to find something that was a little bit more condensed, because I found a lot of the questions on the exams, they pretty much just touch the basics of like methods. So, because I thought, oh my God, I'm going to have to really know every method and memorize them. But, that's impossible. And it's so hard. I mean, for everybody, 1 through 4, they're going to always ask something about BODs. So just, that's a good one to really be familiar with. But yeah, Rich, I was the same way. I got a little bit overwhelmed by, the standard methods. So hopefully I can kind of just round things out. And, but I do like the idea of the KSAs that they've come out with, because that kind of gives you an idea of where your weaknesses are. And I kind of found that to be pretty much right on. So, if you know you're weak in something, maybe go to your supervisors, or try and get on the internet or something that's you can find that's easy to your learn, and kind of try and fill that up. But standard methods is hard. So I'm totally with you there.
And like you said, the KSAs, when you take the test, it does point out where your weaknesses are. I recently have kind of focused my effort on that a little more. But since you brought up BOD, it was something that I had done in my lab for a long time and felt really comfortable with it. Unfortunately, on the test, they present it in such a confusing manner that I kind of got rattled by it a couple of times now. And I understand the calculation, the process, but the way it's presented on the test, it just throws you off.
And that's a big part. What I was going to say too is the way they word things can be really, to me, it was like tricky. And so I went about it as just try and make it easy, and make it rational. Just try and read those questions and read them twice, and just make it so it's rational, because I always thought, oh, it's this is a trick, it's too hard. But yeah, just try and be real reasonable about when you read them, because I noticed that too, you know, and then I thought, well, okay, this is what they're asking, even though to me, when I read the question, it seemed like something I didn't know. So yeah, the way they word things is pretty hard. It was hard for me. So just really read those questions and think, okay, what is the rational questions they're asking me? So, and take time reading the questions so you really get it and don't miss it.
Yeah, I think that's great advice.
Okay, so are we ready to roll through my little slideshow for you guys here?
Yeah.
Okay, let's do it. Okay. So yeah, I don't know what's going to be asked on the test. And I know like for each section, they have 5 different tests. So, you know, it's pretty much every test is different, but the subjects are pretty similar. So, but I do remember questions from different tests, and I don't see why I couldn't put them in my slideshows for you guys. So, okay, is everybody pretty good on how to study and how to study for tests? Because I think Shannon,
Yes.
probably, yeah, I think everybody, it looks like everyone's here has, you know, has done this before and probably has their study skills up to par. I was talking to Amanda and saying that maybe it might be a good idea to put some sort of study session type, I don't know, maybe like a PDF or something that people could download. Because I know for some people, they don't have the skills yet. And it looks like you guys, you know how to study. So I'm not going to go off on that tangent. But so let me just skip around here. Okay, so this pretty good for all grades here is know from talking to people, know these different analysis like chloride conductivity, alkalinity, total and phenolphthalein, pH, temperature, total hardness, turbidity, chlorine residual, free and combined, know what each one of those are. Know like the basics of dissolved oxygen, and definitely be familiar with BOD. BOD, unseated and seated, and the COD, total solids, dissolved solids, suspended solids, volatile and non-volatile, and settable solids. And with the exception of the BOD and CBOD, just know like you can read like the introduction and standard methods, but just be familiar with those really in particular, because I saw those all through each and every exam. And people that I talked to, that, you know, that's pretty much, we agreed on the same thing. So, so be familiar with those different analysis for sure. And let's keep rolling along.
Katie, could I back up just on the phenolphthalein for a second? That is, it's not just an indicator as much as it is the 8.3 pH point too. Is that
Good.
not?
That's a big part of, yeah, that's kind of like the part of it.
8.3.
Yeah. 0.3.
So yeah, be familiar with those different, yeah, what the phenolphthalein does, and like your pH, your different titration points, and it's pretty much kind of like, yeah, be familiar with those. Let me see, I kind of got lost here. Oopsie. Sorry, you guys. So, and then there's other the basic topics like of quality assurance, they're going to ask like the basics on it. It's not like you have to memorize, you know, every little detail on it. Quality control, you know, you probably do this on a weekly basis with all your different analysis, but, you know your quality control, and it's going to be the same what you do in your lab on the test too. I mean, that's pretty standard. And for the one, know some lab terminology. That's kind of big, like those in the back of your study guide. Be familiar with those different acronyms, and know some lab equipment. On that first 1 and 2, they'll ask you some lab equipment questions, but it's pretty much basic things. And the technical terms, that's pretty much back in those back pages of each study guide. Just kind of know, just look at those. I mean, you don't have to memorize each one, but just kind of know the big ones that you've seen before. And definitely like 1 and 2, know your sampling techniques. You know how to sample. If it's bacteria, use, you know, gloves, be sterile bottles. You know, if you have to flame like a faucet or something, disinfect it with bleach. Kind of know those different things when it comes to sampling. And for the 1 and 2, have a good idea of what bottles and preservations you need. I remember the oil and grease with, you know, 1 liter with sulfuric acid, or even I think it's nitric acid. That I saw just about through every test that I took. So, you know, it seems like they tried to base it on, you know, common things that each wastewater lab or even a water company would test for. So it's not going to be like some obscure, you know, it's not going to be like Chrome 6 or something, you know, that you don't do regularly. And definitely know the lab safety and basics of lab calculations, which I think you guys all know that. Let's see. Oops. Okay, the microbiological analysis. This is good for everybody. Just like I say, know your sterile techniques. Does everybody set up microbiology or do any type of bacteria type tests?
I do MTFs at
Okay.
our work, you know, on a regular basis, and Quantitre.
Good, Rich. Okay, good. Because know what the different media goes to, like the lauryl tryptose broth. You know, you need to know that for total coliforms or fecal coliforms. And then it goes into the BGB, and then it goes to EC for fecal, or so know the different, like know those, that lauryl tryptose broth, BGB, and the EC. Be familiar with those, and know that, you know, when you go to autoclave them, you autoclave them for 12 to 15 minutes, because you might see that, and you know, and know what times, like after 24 hours of being in the LTB, you have to check for the gas in the air bubbles, and it has to have the air bubble in that little, I forget the name of it.
Durham tube.
Thank you. Yeah, I know that. So, and then if you don't see any in 24 hours, then you have to wait another 24 hours. So total of 48 hours in the LTB broth, and then you transfer the positives to the BGB. And I hope I'm not repeating this, but it's pretty big on those tests. And then the BGB is used for your total coliforms. And then if you want fecal, then it goes into EC, or you know, and those go for the EC is just a 24-hour test, and the BGB, it goes up to 24 to 48 hours, 2 days. So yeah, kind of be familiar with the media preparation, and they may ask a question. Yep. Question or anything?
She said, and temperatures.
You're going to have a temperature too for the incubation, for the water bath versus incubator.
Good. Yeah. Know those temperature, know your incubator temperatures, and like for Enterococcus, know that temperature for that. I think it's 41 degrees plus or minus 5 degrees, I think. And then the fecal, it's, don't quote me on this. I think it's 40 plus or minus 2. It has to be really right on.
I think it's 44.5 plus
There
or
we go.
minus 0.2.
Thanks, Rich. Boy, you need to be giving this session. Watch out. Okay, and then they're going to give you a sheet with one sheet with formulas on it, but you know what? I mean, I think my grade 4, I finally used that sheet, but for the rest of the grade tests, I never used that sheet for any formulas, and thank goodness I had them memorized. So be familiar with formulas, because don't rely on that sheet. They're not going to give you that many. So a good one to know is the normality formula, and whoopsie. Okay, and then know how to convert like degrees Fahrenheit to degrees centigrade, vice versa. Let me try and center this a little bit more. Oh man, that's not working. Okay. All right. Okay, so we'll go to rounding. And I bet you guys are all familiar with this, but this is a good, I mean, you'll see it throughout. Even on my grade 4, I had one of these questions on there, which it was nice because I knew I got it right. But so just to produce minimal bias, rounding is done as follows. So if the last number is a 4 or less, retain the preceding number. If it's 6 or more, increase the preceding number by 1. If the last number is 5, the preceding number is made even. So here's a couple examples. Like if you have 5.72, that's going to round to 5.7. If you have 5.75, that rounds up to 5.8. Because if you have a 5, you always round your 10th spot to an even number, 1 up. 5.76 rounds up to 5.8. 5.85 rounds up to that should be 5.8, since 8's even number. And then understand control charts.
We have a question.
Yep.
I understand scientific grounding, but in the grade 4, if there's any budget analysis or doing budgeting or fiscal budgeting calculations, counting, you always round up. There to answer this question.
You know what? That is correct. And that's like anything type, business type, always just round up 1 more. And just like Excel does, Excel always rounds it up. So yeah, that's kind of like the gray spot. And you know, if you get an answer like that and you get it wrong, it'd probably be worth it to go back and try and dispute it, because you know, rounding, it's so subjective. Because like Excel, it's going to automatically round numbers up. And then if you're, yeah, so don't be afraid to challenge that if they do, if they mark you wrong or something.
That's a good to bring up, Katie. If you don't mind me interjecting for a second,
Oh, good.
is how to dispute a question on your exam is important, because a lot of people do not flag it and say anything during the exam. They wait till afterwards. And part of the TCP process for evaluating a question, so let's say you had a rounding issue and you felt that it could be one of the 2 numbers depending on the rounding. What would you do is you would flag that question and make a comment formally in the exam. And then afterwards, if you did not pass that exam, it would be reviewed to give you those points back, because there's a lot of people that lose this test by a few points, and every point counts. So take the time after, you know, flag those questions you have concerns about. Go back to them later once you've gotten all your answers. And then the ones that you feel could have more than one answer or are unclear questions or have a rounding issue, take the time to make that comment with your last, you know, time at the exam, because in the end, once you click done, you may find that you needed to have that question reviewed by the TCP committee.
Thanks, Amanda. That's good advice.
That was Shannon, but
That
thank
was
you.
Shannon.
Oh,
love
sorry,
Shannon.
Shannon.
She's my idol.
Thanks, Shannon. Good job.
Thank you, Shannon.
So about control charts. Now, this is probably a good one to go to refer to standard methods and read through it, and just know, like, if you have a point above your upper control limit, that you got to stop your analysis and figure out what's wrong before you start and report it again. So know what those mean. Your upper warning limit, your median. This is a good one to refer to standard methods and read what they have, because it'll be pretty much out of that section. So, and probably you're all doing it in lab, so it's something that's going to be familiar with, which is good, and that makes us good chemists. Let's see. Yeah, definitely know the concepts of precision, bias, and accuracy. So precision is good if it's close together. Bias, I always think of, I love the bullseye. Bias is, I always think you want it right in that center, everything together, group tight. So this one's good good precision and poor bias, because it's so far from the center of what you're trying to really hit. So this one, of course, out here, the underliers are poor precision and poor bias. And of course, what we want is good precision and good bias. So definitely be familiar, with exactly this. If you know this, you're gonna, what did I have back there? So, yeah, the dissolved oxygen, it seemed like I could never get away from this, and I'd never done this, and I don't do it. But I remember they asked me like what the color was when there was no oxygen present, and I never did this method, so I had no idea. So just, review like the basics of it in that standard methods, or something on the internet. I found a lot of good information on the internet that I could actually, understand, because sometimes standard methods is just, it's overbearing. So know that this method removes nitrite. When the flock is white, that means there's no oxygen present. And for the titration of it, you use 0.025 molar sodium thiosulfide. And it's first titrated to pale straw color. Then you add the starch as an indicator, and know these, because these are the type of questions they like to ask you, is like the color of the titration and what type of indicators, and then titrate it until the blue color disappears. So, and for each mil of 0.025 molar sodium thiosulfate is equal to 1 milligram of dissolved oxygen per liter. And definitely know your reagents, like the alkali iodide azide reagent, the concentration of sulfuric acid. And you wouldn't think that they'd ask something like that, but sometimes they get real particular on these type of things. So just know that it's the 0.0021 molar potassium trihydride. I think that's potassium bioiodate, sorry. And that you use 0.025 molar sodium thiosulfate. So this one, it kept coming up through all my tests. So I finally became familiar with it, even though I don't even, run dissolved oxygen. And that's kind of what's hard. That's where the KSAs are good, because a lot of things we don't do, that you're going to be asked about. Whereas the KSAs, it's going to give you an idea of where your weaknesses are. So I like that when they came out with those KSAs. That was a good plan. So I'll go ahead and move on a little bit here. Okay, total solids. Yeah, this is big on all the exams pretty much. So the total solids, it's the residue left in the vessel after evaporation and drying. Total suspended solids, TSS, it's the portion of solids retained on the filter paper and dried. And know what temperatures you dry these at, each one. what incubator temperature and how long. Total dissolved solids, the portion of solids that passes through a 2 micro filter in milligrams per liter. The weight loss on ignition in a muffle furnace, not blast off, is called volatile solids. Settable is applied to material settling out of suspension for 1 hour. Stir gently at 45 minutes, and let it sit for 15 minutes, and that's reported in milliliters per liter. Now that little tricky one with settable solids. So be familiar with like the basics of these, because again, I saw this, you know, these different ones all throughout. And let me get a drink of water here. Okay. All right, back to the bacteria. Know that description of what total coliform is, and that you use lauryl tryptose broth to identify total coliform, and that you, to confirm the total coliform, use the BGB, kind of like what I hit on last time, and then know the incubation temperatures and the time that it sits in each broth. In that fecal coliform, use the EC broth. Here's the definition of total coliform. Oh, I'm sorry, no. But this definition is really good. I saw this a couple times for aerobic, facultative, anaerobic, gram-negative, non-spore forming, rod-shaped bacteria that ferment lactose with gas and acid formation within 48 hours at 35 degrees C. So that's for total coliforms. And I saw that one a couple times. And that, you know, that fecal coliform, the coliforms of fecal origin from the intestines of warm-blooded animals, capable of forming gas from lactose at 44.5 degrees C. Let's see, whoopsie. Okay, MPN, know that it's the most probable number per 100 mils of sample. So that's a good definition to be familiar with. And know that coliform is an indicator organism used to indicate the degree of contamination. It can't quantify it unless you use, you know, like an MPN or something, but they use coliform just as an indicator organism to just indicate if there's other like serious pathogens in that water. So let's see. Yeah, these are good to know for microbiology. Is autoclaving most media and autoclave needs to reach up to 121 degrees and be at 15 pounds pressure for 12 to 15 minutes. And then for dry heat autoclaving, like pipettes or even glassware instruments, a dry heat oven at 170 degrees for 2 hours. And then this is good too. Media can be stored for 1 year. Use it within 6 months of opening. Record date received and open on container. Put when you open it up on container. So, and then media must be sterilized within 2 hours of hydration. You can't let it sit and then put it in autoclave the next day and sterilize it. Actually, I had someone do that, and and the media just it didn't turn out. It actually gathered bacteria. Like when they took it out the next day, even though it's sterilized, it looked like it had bacteria in it. So as soon as you make up your media, try and get it autoclaved within 2 hours. Be familiar with the heterotropic plate count, like how many days it incubates. 2 days usually. And there's a good math question for, I think it's on the 2, about plate counts. So be familiar with it. Just know that CFU means colony forming units. That's a good acronym. And then know how to read the MPN chart. Like Rich, like when you do the Quantitre, you're familiar with how to read the, oh, I'm sorry, no, this is for multiple tubes.
Well, there's an NPN also for Quantitre. It's just got a slightly different, reference to it.
Right, the large wells and the small wells.
Right.
So know how to be familiar with that NPN chart for the multiple tubes. And okay, so there's a couple good exercises, but I'm not going to take our time and actually do them. But this is good. I mean, know like how to calculate the normality of, say, like sodium hydroxide. Actually, I saw this one on the test, and I'm not saying that's going to be on the test, but just know how to calculate, know those formulas by heart, because this is something, I don't even think they put this formula on that little formula sheet they give you. Yeah, like I said, don't rely on that sheet because there's barely any formulas on it, and you're going to be going for the formulas that you use in lab. So, and know how to do this exercise. I'll read it for you in case you're taping this, but what is the normality of sodium hydroxide solution if a 25 ml of a 0.01 normal sulfuric acid solution neutralizes 100 mils of the sodium hydroxide solution? So that's a good exercise. And then, of course, know that N1B1 is equal to N2B2. So just plug in what they give you, what your knowns are, and then that one that you're trying to solve, and that formula will pretty much, get you your answer. So, so here's that problem broken down. So this is a really good formula, and if you know how to work this, it's good for you to just know how to use your math about when to divide and when to multiply. So, and that's pretty much it, thank goodness. It's all about multiplication, division, and adding on these. It's not going to be any calculus or anything like that. So, okay, and these are good questions. So, know what you need to be aware of when sampling outdoors and around a treatment plant. And then know the basics of like your OSHA hazard communication program. And I bet all your labs, you guys, you're all together. So, be familiar with that hazardous communication program because they ask questions on that. And I never really spent any time because I thought, oh, that's, really not anything actually, because I thought it was boring and I didn't want to cover it. I thought, that's not going to be on the test but they do, they have some questions on that. And it's pretty straightforward though. But so okay, here's a good question that you may see or something. So a new lab tech is asked to collect a wastewater sample for metals. The tech grabs a new 1 liter glass bottle from a box of bottles just received from Fisher Science. The tech finds the location, places the bottle under the tap, and collects about 500 mils and returns to the lab. It's quitting time, and the tech sets the bottle on the bench until morning. Okay, first thing in the morning, the tech checks the sample pH and temperature and logs the sample in for analysis. Well, I'm sure you guys can, figure out, what he shouldn't have done and probably what he should have done. So, just know what you're going to be what you're going to be analyzing for, and then think and look up what bottle that analysis is required of. And so for the metals, he should have had a plastic bottle preserved with nitric acid. And so yeah, know what, be familiar, especially, well, the grade 1, yeah, Shannon, be familiar with like bottles and preservations, because I remember that was pretty big on the lab 1 test. And
Okay.
yeah, with hold times. So I mean, it's not like, and it's like the common analysis. It's not anything, really far out there, things that we don't do on a regular basis. So
Okay.
Yeah, that was big there. Bottles and preservation and what analysis they coordinate with. And then,
Okay. And
then,
chain of custody.
mhm. Was there a question?
Jen was mentioning chain of custody, and
Yeah, there's no possession of the sample, so it could be considered invalid as
Good.
well.
Good. Yeah. Know that you got to fill that, you got to log that sample in, fill out that chain of custody. And Shannon, be familiar with that, how to fill out a chain of custody. And when you log it into, say, your logbook or something, just know that,
yeah,
be familiar with that too. Just the different outlay of how you log something in, like the time you sampled it, where you sampled it,
okay.
the nature of it. So I won't go too much into that, but
Katie, in the whole time in standard methods, make sure you ignore the suggested, only go with the regulatory.
yeah, definitely. Either go to like EPA online or the CFR 40 and look up hold times, because I found where standard methods just, go with what is regulatory. Like Rich says, that's a good point. Thanks, Rich.
And they have a whole sheet on the EPA bottles and hold times and things. So that would probably be sufficient.
And definitely for grade 3, you need to go by the EPA sampling and preservation requirements and holding times, not standard methods.
Good. Yeah, that's EPA. Good going. I should actually put that in my study slides here. Let me just make a note. Yeah, if you can, go to the CFR 40, of different analysis and such, because it's pretty right on. Yeah, I didn't really have too much good luck with standard methods. I mean, of course I follow it, but it's not easy reading, that's for sure. Yeah, and if you guys have input too, I appreciate this. Thanks, Rich and Shannon. This is good because I'm learning too. Yeah, just any problems with the protocols above, but I think you guys have a good idea of that. So we'll go ahead and move on here. Let's see. Okay, and just touch, I'm just going to touch a little bit about different ways of learning, because in the study guides, I know there's a big section on it, but you guys are familiar with learning and how you study and what works for you. Like for me, I know that if I can actually read it and say it out loud and hear myself, that's how I study better and learn things. I know some people, if they see it, it works for them, or if they hear it, which is a good thing. But we all kind of have our different ways. My son's lucky; he can just read something, and it's set in his brain. And I'm kind of jealous of him about that. But so just kind of know, what works for you. If you have to listen to it, or if you have to see it. And I'm actually the type where hands-on is like the best thing. So just kind of be aware of how you learn good, and and rely on it, and trust yourself. Okay, visual learners. I'm going to skip through this. I don't think we need this. Yeah, so for grade 2 people, so know that grade 1 study book. And the study books are pretty good, except for like the 3 and the 4, because 3 and the 4, they get more real particular about, what instrument you're going to use. There's lots of questions, and that gets pretty hard because a lot of us don't use a lot of different instruments. Like I don't do any organic analysis in my lab, so I don't have, the GC or the mass spec, and they're going to ask questions on that. But
we had a question for you.
Did you take your grade 4 prior to 2015 when we had the old study guides, or did you take it when they had the second edition? Because there's a huge difference between the tests.
Okay, all right. So I think I took it in 2015. So I just missed that one. I just missed
Okay,
the
so
grade.
that's the old study guide.
Oh, good, because
Okay.
I hope, yeah, and they were supposed to not, ask so many questions about different instrumentation, and I hope they did away with that, because I always kind of felt it wasn't fair, because a lot of us, we don't have a mass spec, we can't afford a mass spec in our labs or anything, and so yeah, I'm not familiar with the new updates on the grade 4. I should get in with that. So hopefully they did just kind of more, hopefully they made it more like what we do actually day to day or something. I'm not sure.
I found something that was really helpful for me because I've never done DC either. I went onto YouTube and found some great videos on people just demonstrating how their instrument works, and it was very, very helpful, and also with atomic absorption as well.
Good.
So it's a good tool on YouTube.
Are they still asking about
Yes.
atomic absorption?
Yes, they still ask about atomic absorption.
Are you serious?
Yes.
That was Jen and Ron.
Sorry.
I'll confirm that too on the 3 they ask.
Yeah, and because I had a couple of my friends that just took the 3 and the 4, and they said that they're still, asking questions about the mass spec and the GC, and I think one person they asked, you had to actually identify parts to it, which I don't think that's pretty unreasonable. But that's a good thing. Get on YouTube, because what I did is I went to a lab that I subcontract my organic analysis and my metals to, and I asked them if they could just take me through like HPLC and the mass spec. And I got to go in their lab and see the equipment, and that helped me, because I'm more of like, if I see it, and if ideally if I could do it myself hands-on, I learn better. But no, YouTube, that's awesome. That's great. Good recommendation.
Maybe quick question. Do they have I know they have AA. Do they test IC?
Yes.
IC mass spec?
I didn't hear that question, sorry.
He was talking about ICP.
Yes.
ICP, AES, they can ask any of those questions. I had at least 2 or 3 questions on ICP for my grade 3 last year.
No, they were big on that, and the mass spec too. I mean, even from my tests, they were. So, and what else? Yeah, we'll get a little bit later. I'll touch some things on that, which I kind of gathered information on the 3 and 4 from people that have recently taken it. And so later in the slideshow, I'll kind of get into that too. So let's go back to grade 2. So know that grade 1 study guide. And a lot of the questions actually come from the study guides. I mean, they might just, like for the math, they change some of the numbers, but I found most of the time, if I knew that study guide, it probably helped me pass my tests, really. So know those study guides real good. And so grade 2, know everything in the grade 1 study guide. Be familiar with
Jen has a tip real quick.
Sure.
I found that there were a couple questions from the glossary of definitions too, so know those if they're easy points to get on your test.
Yeah, be familiar with, like, when I went through those acronyms in the back of the book, the definitions, I would look for stuff that I had kind of seen before, because I tell you the truth, you guys, I didn't have a lot of time to study for these, and so I tried to just it's not good, but I tried to do bare necessity. And so when I looked at those acronyms, I thought, okay, well, which ones have I seen somewhere before? And then I familiarized myself with them. And that kind of worked, because I think they take things that, we all probably should see through the course of our lab work. But there's going to be some of those on there. So for grade 2, be familiar with ammonia, nitrite, TKN, and phosphorus. Phosphorus was a big one for me, because I'd never done any of it before. And there are questions: fluoride, oil and grease, sulfate, and the surfactants, MBAS. like we went before, we talked about the precision and accuracy and bias and sampling protocols, temperature conversions, know just a little bit about bioassay, know general principles and practices of the UV visible light spectrophotometer, the AA and the AA using cold vapor for mercury, the AA using gaseous hydride, and TOC and ISE probes. Just know some of the basics because you're going to see those things popping up. And if we're not using them in lab, it's kind of shocking. So, and that's good. Get on YouTube. That's a great idea. You know, see how an AA works or TOC. So let's move on here. Yeah, this is for the AA, know the introduction to metals. So know this, how to sample for it, the preservation, and like the precautions of what you don't want to do. QC, preliminary treatments, like what you preserve it with. And then know in general what metals can be analyzed by the atomic absorption. And this is a good one to refer to. Standard methods is read like the introduction, SM 3111A, and be familiar with the section titled Principle. Know that gaseous hydride is applicable to, what is that? Arsenic and selenium. I saw that I had that one 2 times on a test. Cold vapor, that's applicable to mercury. So just, yeah, let's see what else we have. Okay, these are ones that, I mean, from grade 2 on up to 4, you might see these. So for total organic carbon TOC, I went to standard methods 5310, but if you can, you know, find something online that makes it easier, I definitely suggest it, because standard methods, it was really hard for me to get with this one. Read the intro, be familiar with combustion infrared, and I saw that one on the 3 and 4, and the persulfate UV oxidation methods. There's a couple different methods for just TOC. So be familiar with the introduction to each of them. Know what an ion selective electrode is. You can find it in standard methods, 4500s. You can use it for pH, fluoride, nitrite, nitrate, and ammonia. Whereas I think everyone's using probably like maybe a GC to run those analysis. I think just small labs like myself use the ion selective electrodes. I don't think many people use those anymore, but unfortunately I still do. Okay, and then more getting off the beaten track of what we do in lab, be familiar with like basic wastewater treatment processes, more of like what the operators do, just because they may ask something when a plant gets upset, you know, what you're going to be looking for, or if it's digester, you know, your pH and your alkalinity. Know the basics of a treatment plant, kind of what takes it, you know, the process of it. And I know in labs sometimes we're far removed from the operators, but I'm pretty sure probably lab managers, they actually have to help, you know, the operators when things do go wrong and suggest things. So be a little bit familiar with what the operators are doing. And so, okay, just, yeah, read the introductions. And if you need to summarize things on 3x5 cards, if that helps you, it kind of helps me, you know, if I can see it again, if I write it down, see it, it's good. And I can take them with me, you know, if I'm doing something, I can just pull them out. Are you guys about ready for a break?
That was a yes.
Do you want to take a break?
Everyone's calling, but I have you on mute.
Okay. So maybe let's take a 10-minute break. I'm going to get some water and we'll be back, say like 10 after 10.
Okay,
Sounds
thank
good.
you.
Okay, good. All right. Thanks.
Observe.
I wanna come and get retrained on how to do this.
You know what? Yeah. You know what is it? That this is the one old music.
Okay. Let's see. Is everybody back? Please move this one.
Yep.
Yep, we're here.
We're all here.
Okay. All righty. Okay, so let's go on to the next slide here and know a little bit about, bioassays, the fish toxicity. Know that there's two types, acute toxicity and chronic toxicity, and that both run a reference toxicant test with sample. Actually, I think that's out of one of the study guides, and it could be a test question. Know that the acute can be flow through or static renewal and can use adult or larval fish or other organisms such as algae. And it's mostly 96-hour tests that we'll see, like for what we do, results as lethal concentration is 50% or percent survival, and it measures mortality. So know that about acute testing. For chronic is static renewal. It has a critical life stage. Results are in toxicity units, and it's run as a series of dilutions. An example, like a 2.5% solution or 5% effluent, and it has control water also. So I don't remember anything like on the grade 1. They didn't test anything about bioassays, but it's just good to know the two different types, acute and chronic. So let's see. And good old BOD, memorize the BOD formula. I highly suggest it. Just know the BOD. Seeded, unseeded's real easy, but know the seeded BOD formula. And even the grade 1, just be familiar, because you can see that they like those BOD problems. And then a little bit more about BOD, that the GGA standard, the true value is 198 plus or minus 30.5. So it has to be, your GGA has to be in that range. And if it's out of range, then that's when you can't report those results. If your GGA is out of the range, you can't report those results. So it's not anything about your blanks or it's GGA. That kind of like verifies the BOD analysis. So know that. That's why I keep saying this again and again. Yep.
Hey, Katie. This is a lot of really good information. Are you going to make this presentation available to all of us that attended?
I'm sorry, what was that? Was I making?
There's a lot of good information on these slides. It's hard to write everything. Are you going to make these available for everybody who attended?
Yeah. You guys can have a copy of the slideshow.
Thank you.
I guess
really good.
Oh, good. Oh, thanks. All right. I hope so. I hope you guys. But like I say, I've talked to a lot of people recently who've tested like through 1 through 4, because I didn't want to get on here and, or give a class and, lead you guys in the wrong direction. But pretty much from what I've gathered, and I've done a lot of gathering, is if, these major topics everyone has seen. So, I'm trying to just make it easier for you guys so you're not like going through standard methods or you're not getting off on a tangent, because I got off on tangents, and I wasted time studying things that probably none of those tests ever had on them. So this is I really feel it's a good direction. And I'll put up my email, and then just shoot me your email, and I'll send you a copy of these slides, and we'll do it that way. Okay, so be familiar with the BOD parameters, like the blanks. They must have like a less than 0.2 milligrams per liter depletion to be valid. Even though if those blanks go over, if your GGA is within value, those results you can still report. So everything depends on GGA value. You know, your blanks, they can be over or whatever, but it's just an indicator that you need to figure out, you know, if your BOD water is not up to par or something else. And then the BOD must deplete by at least 2 milligrams per liter DO, and you must have at least 1 milligram per liter remaining. The DO uptake is seed control. It needs to be between 0.6 to 1 milligrams per liter. And if you have samples that are chlorinated, you need to dechlorinate them first. You need to adjust pH of your samples. If they're out of range, they need to be within 6.5 to 7.5. Sometimes my influent runs a high pH and I have to adjust it down. Sometimes it's like in the 8s or so. So I need to, you know, use a little bit of sulfuric acid and get that in the right range before I set up. And then just know the hold times. Ideally, you need to set up within 6 hours of collection. They do state though, you can wait 48 hours, but before you set up.
Yeah.
And your DO water, it needs to be, the oxygen needs to be, okay, if your DO water is greater than 9, you need to shake it up and, or aerate it, and it needs to be set up at 20 degrees C. So what I do with my BOD water when I make it up, oopsie, darn it, okay. Okay, when I make my BOD water up, I usually let it aerate, run air through it to help supersaturate it. And that brings my DOs down to, say, usually about 8.3. And you want your DOs for your BOD water to be between, you know what, and it's on the top of my head and I'm embarrassed because now I forget, but I think it's, I apologize for this. Okay, so just know what dissolved oxygen your BOD water should be at. I'll put it that way. I don't want to give you any wrong information. Okay, back more on total coliforms. So I might have reiterated. Let's see, double. Okay, so double confirmation, use the BGB for total coliform detection, and use the EC broth for fecal coliforms. And this is for the completed phase. And it's in standard methods, but I think this kind of breaks it down easier for you. So you need to streak an LES endo agar plate from the BGB positive tubes. You need to transfer the discrete colony to an agar slant and also into a single strength LTB tube at the same time. You incubate that overnight. If there's gas in the LTB tube, it's positive, and then you use the corresponding slant growth to make your slides. Okay, this is like a general breakdown, so definitely either get online, or if you have to use standard methods, you know, read through what the completed phase is, because it's kind of important. Make a light emulsion slide, air dry it, fix it, and then it's a gram-negative stain. So there's a procedure for gram-negative stain. I don't know how familiar everybody is with it, but basically you stain with crystal violet. You let it sit for a minute. You take it out and you rinse it with water. You apply Lugol solution, let it sit for a minute, and then you rinse. And then you do another rinse with acetone alcohol. I think it's for like 30 seconds or until you see it kind of go clear. And then you counter stain with safranin. And then you rinse it again with water and you blot it dry. And then you look for the characteristics of what a gram-negative stain should be. And so the gram-negative stain, you'll have the blue rod shape, and the gram-negative are going to be red. So kind of be familiar, even though, you know, I don't know if a lot of people, anyway, I have to do the MPN by multiple tube for total coliform and fecal coliform. And I remember seeing the completed phase, you know, on the test. So I was happy that I knew, you know, the basics about it. So it's completed phase for total coliform. So kind of go over that, and then what a gram stain is, and then what they look like. Like gram stain positive is blue color, and the gram stain negative is red. So be familiar. Okay, here's some good questions. So what organisms does the test measure? Like for, you know, total coliforms or fecal coliforms, that's kind of basic. What's a brief summary of the method? Kind of what we've been going through, like for the lauryl tryptose broth for total coliforms. What media is used? What type of labware is used for the method? Like culture tubes, pour plates, that type of thing. Incubators, what temperatures? If you have to dilute the sample or if it's used at full strength, and how the media is inoculated, like serial dilution, using the sterile loop transfer, or streaking on agar. So just be familiar. There's a lot of micro things, and they, you know, you just don't know what they're going to ask about the microbiology. So kind of have a good feel for that. Okay, here's a good exercise. I'll read it to you, and then you guys can do it on your free time. So a 200 ml sample of chlorinated effluent is analyzed for total chlorine residual using an amperometric titrator. The potassium iodine and pH 4 buffer are added to the sample. 5 mL of the 0.00564 normal phenyl arsine oxide are added, or POA, are added to the sample and back titrated to the endpoint with 0.6 mils of 0.0282 normal iodide solution. What's the total chlorine residual? And yeah, definitely let me email these slides to you, because this is a real good question. I mean, I said it a few times, so they like to do that. They like the math in it, and it's a good exercise. It really is. Okay. So here's the formula for it and the breakdown of it. And if you guys can do this math, I mean, that's probably as complicated as it gets, which is good. So that will be in the slide for you guys. Okay, then back to like the sample receiving log and more like the chain of custody. It kind of sounds like everyone's doing that already. So I really won't go off on that. But just, you know how you log your samples in, what parameters you log, and
Sample name.
what was that?
Sample name.
Yeah, like the name, the date that it was taken, who took it, the nature of the sample, if it was a grab, composite, if it's intact, the analysis.
How many bottles? No.
I'm sorry, what was that?
I was just saying, I think from Riverside, I remember we'd have to log in how many bottles we had for each sample. Like
Yeah, exactly. It's particular.
1 to 5.
And let's see, where's a good example of like a login chain of custody? Actually, I think the EPA, they have like what they recommend. And so that's what I go on my lab, just what they want, you know, and ELAP, they'll let you know too. I'm sure everyone's kind of gone through that deal. And yeah, know how to fill out a chain of custody and how to log it into your laboratory. So it's, you know, that's a legal form of how you follow that sample from the day it enters into your lab till, you know, the day the report goes out. Were there any questions? Okay.
Nope.
All righty. So then you know, here's an example of, you know, what the receiving log must contain, and this is EPA. So date and time sampled, date and time received, sample collector, their initials, sample description, if it's an influent or, you know, if it's effluent, reclaim, the nature of the sample, if it's a grab sample or a composite, analysis to be performed, preservatives used to on that sample, and then whatever your lab, however you ID your samples. Like for me, I use the year, the date, and like my sampling site number. So whatever your lab uses to identify it. And okay, we'll move on here. Oh, this kind of, yeah, but this is a good question. Of the 4 possible outcomes, you guys might have seen this on some of the 2 and 3 exams, but of the 4 possible outcomes of bias and precision, which condition is correct? A, low bias, low precision. B, low bias and high precision. C, high bias and high precision. Or D, high bias and low precision. So can anybody tell me the answer for that?
B.
Good. That's what I want to hear. All right, you guys are staying with me. Thank you. Okay. Whoopsie. Okay, that shouldn't be there. All right, I need to go through my slides again here.
Isn't that right? Wasn't it low bias and high precision?
It should be low bias because they're all grouped together, and high precision, we want them all together. So whoops. So it is B, I'm sorry. Okay. So is that kind of clear? Sorry, I messed that up on that last up here. So it's,
Yeah, I think it's perfect.
yeah, low bias, you want everything grouped in that tight little center, and high precision, close together as can be. Okay. Yeah, a little bit more on total TOC. I saw a lot of that on 3 and 4. I've heard a lot about it, that people get asked that just recently. So be familiar with the total organic carbon, and that there's 2 methods: the UV persulfate oxide, that's used in drinking water industry because it's more sensitive than combustion. So increase in solids, increase in bias. The combustion infrared, it's used for wastewater and stormwater. It's a more complete oxidation of TOC in solids, not as low detection limits like for the UV persulfate for drinking water. Know the different forms of carbon. There's total carbon, total organic, inorganic, carbonate, bicarbonate, and dissolved carbon dioxide. A little bit about the AA. Okay, the AA spectrophotometry measures a wide range of elements. The digested sample is aspirated into the flame of the instrument and atomized. Light of a specific wavelength for a specific element is shown through the flame, and some of the light is absorbed by the atoms of the sample. The amount of light absorbed is proportional to the concentration of the element. So everyone that I've recently talked to said that, you know this definition of AA because you might see it. Yeah, on the practice guides, do those math problems and just know them, because I swear, like when I've talked to people, it's like you see the same question, but just the the numbers are changed. So, that's something easy that you can figure out and have ready for when that, when you sit down and take that test. Let's see, I have a couple more things, and actually, here's some good information guides. WEF, I don't know if anybody's a member, but they have a good site where you can take different quizzes, and they have information on their site, which is good. Sacramento, the University of Sacramento, they have some really good online training, underwater programs. They have some good good sessions that you can.
I'm in a conference video call right now. Anyway.
Was that you, Rich?
Okay, I should be done Okay, right.
Actually, I'm wrapping things up. Maybe I condensed this too much, but I'm about done, actually. let me, okay, ELAP site. I didn't find that one too helpful. I shouldn't say that, but in balancing equations 3 and 4, this is a really good website of how to balance equations because, you know, I'm really poor at it. And so I use this. This was helpful. And 3 and 4, you could see, you know, you might see a question on balancing equations. And that was something that was hard for me. Now, at first I thought, oh, I know I'm going to get it wrong. So I kind of wrote it off. But, you know, just kind of be up on it. If you got it already under control, you're doing good.
YouTube was really good for that too, Katie.
Okay.
Tyler DeWitt on balancing equations.
Okay, good. Yeah, because I struggled. And this is good for 3 and 4. If I can say one last thing, no EPA method 608, 611, 624, 625 pertaining to the new method update rule. You know those, be familiar what instrument uses those. And also this question, everyone says, know that a Caderna Danish concentrator, what that is and what it goes to. So know that terminology and what it is, because that's a good, that you might see that. It's a good possibility from what I'm hearing. Let's see, this book is awesome. I don't know if you can buy it or not, but God, I mean, I found so many, I think, questions from each test was out of this book. It's called Lectures in Wastewater Analysis and Interpretation. And it's a blue book by Roy Keith Smith, PhD. The publisher is Genium Publishing. This book was awesome. I mean, it put standard methods back on the shelf. It's easy to read. I think it costs like $66 though. I borrowed one and it's really good. I want to get one just to have for my own reference. So that, okay. And then this is what I remember from, actually this was on the grade 4 test, and I only deal with drinking water. And, you know, I have my biochemistry. I worked in toxicology for years and years, and Scotts Valley was my first wastewater plant. I do work for the water district for water samples, but I'm not familiar with water samples per se. So on my grade 4, there was major cations in water. They're asking me, you know, what were the major cations in water? And, you know, I had a guess, but I had no idea. And then there was another question, major anions in water. And, you know, I was just guessing. So if it helps at all, and I know I'm not supposed to give you questions because you don't know if they're going to be on there or not, but I remember I was disappointed because, you know, if I knew there were going to be questions on water, drinking water, I would have kind of boned up on stuff. And this is a good question, the TDS to EC ratio in water. That one, I made a good guess. And then know the bacteria that's used in the water suitability test, and then what the parameters are for it. And we already touched base. Well, bacteria used to QC total coliform. That's a good one. Okay, then here's like more of a process control question that they could ask you about a treatment plant. And actually, this is what I remember from a test. And I know I'm not supposed to, you know, tell you what's on a test, but I remember this question. It was, what test is used to see how clear the final product is in a wastewater treatment plant? And they give you like BOD, reduction, TSS, and settable solids. And the answer is total suspended solids. You know, how many solids are still remaining. So if it's high, then you know that the water isn't, you know, as good as it should be. And I think that's pretty much what I have condensed in. What did we do? Like an hour and a half. Okay. Well, I could have put in more things.
Katie, could I cut in on that last one? What test used for final clarity of the water?
Yeah, good.
It was both BOD and TSS, is what you said?
No, it was TSS.
Because for our CWIX reporting to the State Water Board,
Mm-hmm.
We have to report both BOD and TSS.
Yeah, I do too. You know, research this question out because, you know, I could be wrong, but
Uh-huh.
yeah, know that one because I had to think about this. And
Okay,
yeah,
but for the test, it's primarily TSS is the concern.
yeah, but you know, but question it, look at it and see. And,
Okay.
but you know, I'm with you, Rich. I have to, you know, I thought it was BOD at first, but for, yeah, CWIX, I have to do BOD and TSS too. So, but yeah, you know, check that question out and just get a really good firm answer on that because these were questions that I was, you know, I was really unsure of. So if I can pass it on to you guys and if it helps, I hope. So anyway, so this was a condensed version today, you guys, and I hope it helps you out. Shoot me, let's see, I don't think I put up my email address, did I?
I don't think so.
Okay, well, you know what? I'll have Amanda get me you guys' emails and I'll send you my slides.
That'd be great. Really appreciate it, Katie.
And Rich, you know, I think you're set. You could probably be teaching this, doing this. We need people to teach, you know, because it's no fun going to a study session and just seeing the study guide. You know, it's not going to get us anywhere. So if we can put on what our knowledge of, you know, what we've experienced and what we've gathered from other people, I think that's, you know, pretty helpful because I don't think a lot of us have, you know, all the hours in a day to be sitting and studying for cert tests. So, you know, that's my main point is to really kind of touch on what you might actually see on these exams. So you guys, I'm going to let you go and I'll get with Amanda and I'll email you my slides and I hope it works out. You know, give me some feedback. Let me know if I'm doing any good or certainly let me know if you pass your exams because I don't want to be doing this if I'm not helping people. So,
I think you did great, Katie. Thank you.
Hey, thanks, Rich. All right. Well, and good luck, everybody, because I know these are always so stressful and, you know, and we have other things going on in our lives. We just need to make it simple and get through it. So, thanks,
Awesome.
everybody.
Thank you.
Thank
All right.
you.
Thanks, you guys. Have a good weekend.
You
Amanda,
too.
thanks. Are you still around, Amanda?
Yeah, just as a wrap up, we can connect via email so that we can share this recording with everyone and so that we can share your slide deck with everyone. And then also so they have your email address if they wanted to ask any follow-up questions as they're processing information. Maybe a question will pop up and they'll be able to email you. Yeah.
Are you going to put it up?
All righty. Well, thank you, everyone. And again, thank you, Katie, so much for donating, volunteering your time and your expertise. It means so much to us, and we're so grateful. Thank you, Katie.
Oh, thanks. And I can be better, you guys, so I need help too. So any feedback's appreciated. But yeah, good luck and thanks so much. Thank you, everybody. Have a good
All
week.
right.
Right.
We'll see you on email.
Bye.
Bye, everybody.
Bye-bye.
And thank you very much, Amanda, for setting this up.
Oh, my pleasure.
Yeah, that was
My
great.
Thank
Good
you.
job, Amanda, and Shannon too. I mean, I'm really impressed. You taking steps to, you know, be able to help out the lab more and getting your knowledge up to speed. That's above and beyond. We need people like that. That's really great. That's good.
Thank you, Shannon.
Yeah, good job. I think maybe they signed out, but yeah, thanks, Rich, and enjoy San Luis Obispo.
All right, thanks.
Okay.
Okay. Have a good weekend, everybody.
Thanks. Okay, bye-bye.
Bye.